We now make a hard pivot in the course material from organic chemistry to biochemistry. Biochemistry is the chemistry that is focused on the molecules of living things. Turns out that aside from water, organic substances make up about 5/6 of the remaining mass of our bodies. And so while our focus in biochemistry will be on specific types of organic molecules like proteins, lipids, carbohydrates, and nucleic acids, these molecules are all organic molecules, and they're defined by the types of physical and chemical properties that we've been talking about throughout the last chapters. Biochemistry at the survey level is a lot more about giving you exposure to the types of biomolecules in our bodies and cells, and a lot less about a discrete set of skills than what we saw in organic chemistry. So while we'll still have a few discrete skills like that, the reading guides and noteworks will be a much better and more complete preparation for the exam than the homework for the remainder of the semester. We see carbohydrates show up all over the place. It's tempting to think of carbohydrates as limited to things like bread and pasta. We'll see that carbohydrates make up a portion of a wide variety of foods, and how the types of carbohydrates in other foods are often more important. Carbohydrates also play an important role in the world around us, giving the structure to the tree trunks or differentiating between gram-negative and gram-positive bacteria, and even serving as part of our immune response to those. At the most basic level, carbohydrates are built of units called monosaccharides that have a single aldehyde or a single ketone and an alcohol on all the other carbons. Monosaccharides with an aldehyde, like the one shown on the left, are called aldoses, ald for aldehyde and the ending -ose being used for most carbohydrates. Monosaccharides with a ketone, like the one on the right, are called ketoses. Notice that the ketone on the right is a ketone, not a carboxylic acid, because the carbonyl, the carbon double bound to oxygen, is on a different carbon than any of the alcohols. This makes it a ketone with an alcohol, not actually a carboxylic acid. We'll see that at the end of this lesson that individual monosaccharides can form rings that are hemiacetals with their own aldehydes or ketones, actually at the beginning of next lesson. We'll see that at the beginning of the next section that those monosaccharide hemiacetal rings can be linked together as acetals that make up disaccharides. That is a carbohydrate that has two units tied together by an acetal, or even longer chains like other oligosaccharides, chains of carbohydrates up to 10 units long, or even polysaccharides that can have thousands or even a million monosaccharide units linked together. In addition to classifying as aldose or ketose by functional group, we also classify the carbohydrates by length, using the same Latin prefixes we used to indicate the number of identical side groups before: di, tri, tetra, penta, and hexa. So a 3-carbon carbohydrate is a triose, a 4-carbon carbohydrate is a tetrose, 5 is pentose, and 6 is hexose. We can combine these systems. For example, the molecule shown on the left, D-galactose, is an aldose, which we can tell from the aldehyde on top. It's also a hexose because it has 6 carbons. Make sure you can count where the 6 carbons are coming from on this molecule. If you're counting 7 or 8, you're probably counting the hydrogen at the top as a carbon, it's not, or the OH at the bottom as a carbon, which it's also not, or maybe you're counting both. We can refer to these together, the aldose and the hexose, by calling the whole molecule an aldohexose. So that refers to both the functional group and the length. Pause and try to use this pattern to identify the type of carbohydrate the D-glyceraldehyde is, and the D-cyclose as well. D-glyceraldehyde is an aldose on account of the aldehyde at the top of the molecule. It's also a triose, having only 3 carbons. Together that makes D-glyceraldehyde an aldotriose. D-cyclose is a ketose, since its carbonyl has carbons on both sides. It is also a hexose on account of having 6 carbons. Together, it's a ketohexose. You may have noticed that there are a lot of dashes and wedges in the carbohydrate structures we draw. That's because the differences in 3-dimensional orientation actually represent different stereoisomers. That's molecules that have the same connectivity, but necessarily a different orientation in space. These small differences make an enormous difference in how molecules behave in our bodies. The molecule shown on the left is a 3D structure for D-glucose, one of the main forms that dietary energy is carried through our blood. The molecule on the right is D-galactose, a molecule that's part of many diets as the disaccharide lactose, as a component of that disaccharide. The only difference between glucose and galactose is the direction of the oxygen, the fourth carbon from the top. So we're talking about this oxygen. On glucose, the oxygen is pointed forward out of the screen, indicated by a bolded wedge. On galactose, the oxygen is pointed backwards, away into the screen, indicated by a dashed line. This small difference gets amplified biologically, where these two aldohexoses that look very similarly will serve different roles biologically. We call molecules that have different possible 3D structures chiral molecules, and the different versions are called stereoisomers, molecules that differ only in the direction of their bonds but never the connectivity. We can tell when a molecule is chiral when the molecule and its mirror image couldn't be lined up if you put them on top of each other. We'd say that it has a non-superimposable mirror image. A really easy example is your hands. If you line up your thumb with your thumb, your pointer with your pointer, and so on, every finger bends in the opposite direction on your left hand than it does on your right. If you tried to line up your palms so that they were in the same direction, your thumbs would be on opposite side. So they're mirror images, but they are not superimposable. This can be true in molecules as well. The molecules shown in the top right are two stereoisomers of glyceraldehyde, our simplest carbohydrate, or our simplest aldose anyway. There are mirror images of each other, but there's no way to line up their bonds. In the molecule in front of the mirror, the OH in the middle carbon is pointed out towards you, indicated by that bold line again. But then in the mirror image, the OH is pointed away into the page or into the screen. We call the carbon that can have groups pointed in different directions in order to make a different molecule a chiral center. In this class, chiral centers always occur when an atom, usually a carbon, has 4 different groups attached. So practically, how do we tell if a molecule is chiral? We'll look at each carbon to see if it is a chiral center. If a molecule has a chiral center, it usually is a chiral molecule, and it certainly will be in this class. We'll try the molecule on the left, which is threonine, an amino acid, and amino acids are the building blocks for proteins, so it's a pretty relevant type of molecule. Starting with the carboxylic acid carbon, which is this one, we'll check to see if it has 4 different groups. Since carbon can only have 4 different bonds, anything with a double bond necessarily has a maximum of 3 unique things attached, since one of them is attached twice. So then we'll move on to the next carbon right here. This carbon is connected to a nitrogen, that's one thing. There's also a carboxylic acid that's attached, that's 2 things. There's also a 2-carbon chain with an alcohol over on the left side, that's 3 things attached. And there's also a hydrogen, that makes 4. If you don't trust me about the hydrogen, remember that line angle structures don't show hydrogens attached to carbons. So if a carbon has 3 unique groups that are shown single bound, the unshown hydrogen will always make it a chiral center. So 3 things that aren't hydrogen and no double bonds would mean that the 4th thing makes it chiral. So that's our first chiral center here. This molecule, like most amino acids and therefore most proteins, is chiral. This amino acid also has another chiral center on the next carbon though. We get that from the OH, which is one group, a CH3 that's above it, above this carbon, and that's another group. On the right, we have a carbon chain that has both an amine and a carboxylic acid. That's a 3rd group. And of course, the hydrogen that's not shown to make 4 different things attached. So that carbon is also chiral. The 4th molecule, or sorry, the 4th carbon in this chain right there is a CH3. Since there are 3 hydrogens that are each no different from the next, that carbon is not chiral. Now look at the next molecule, which is a carbohydrate. We'll start with the aldehyde at top and try to identify whether each carbon is a chiral center. The aldehyde carbon itself has a double bond to oxygen, which means that it can't be bound to 4 things and therefore can't be chiral. The next carbon is bound to the aldehyde carbon that's above it, and it's also bound to the chain of 2 carbons with alcohols on them below. It's also bound to an OH directly on the left. That's a total of 3 things that are single bound, but none of them are hydrogen. The unshown hydrogen will make a total of 4 groups attached, and therefore that is a chiral center. Now you try the third carbon down in this carbohydrate. Is it a chiral center? The third carbon down has a 2-carbon chain above with an alcohol and an aldehyde, and it has a 1-carbon chain below with an alcohol only. Even though the first carbon above or below the carbon we're looking at looks similar, the chain length and functional groups further out are different. And that still matters. It doesn't matter how far out the difference is. If they're different, it could be chiral. There's also an OH and a hydrogen on that same carbon for a total of 4 different groups and making another chiral center. Now try the 4th carbon down in the carbohydrate. Is it a chiral center? Since only 2 groups are shown, the carbon chain above and the OH below, the other 2 bonds to this carbon must be hydrogen. With 2 bonds to the same thing, hydrogen, this carbon cannot be chiral. Now try the final molecule, which is a fatty acid. Try each carbon and try to identify if there are any chiral centers on the fatty acid. The fatty acid has no chiral centers, as the carboxylic acid carbon only has 3 groups bound, and every other carbon has at least 2 hydrogens. In addition to a little bit of practice identifying chiral centers, hopefully this underlies that chirality is really important in carbohydrates and proteins, but less so in fatty acids, which are a major component of most lipids. We talked about our hands as being good examples of chiral molecules that are stereoisomers. Again, that's isomers that differ only in orientation. Our hands are an example of a specific type of stereoisomer called an enantiomer, because our hands are mirror images of each other, but are not superimposable. Enantiomers, which again are always mirror images, have nearly identical properties in most situations. So imagine holding a ball in your left hand, and imagine holding that same ball in your right hand. Your fingers can clasp around the ball in equivalent mirrored ways, allowing with your interactions with the ball to be the same. That's because the ball is not chiral. Now imagine a structured glove like the one that's shown, or like a baseball or a softball glove. There are chiral versions of these gloves, either right-handed versions or left-handed versions. If you try to use the right-hand version on a left hand, it will have completely different properties. This is important because most of the organic molecules in your body are chiral, and so they will interact differently with other chiral molecules, just like a softball or baseball glove would interact differently with your right or left hand. It will matter which mirror image of a molecule we have. There's another type of stereoisomer relationship that is not a mirror image, but still has the same connectivity. This is like the glove on the right compared with the other 2 gloves. Since all of the hand is still connected in the same order, it's still a stereoisomer. But since some of the fingers had flipped directions, but not all of the fingers had flipped directions, this glove isn't a mirror image of either of the gloves on the left. We call molecules where some chiral centers flip, but not all, diastereomers. These are different than enantiomers because they'll have completely different interactions with each other, just like this hand would have completely different interactions with a ball. As a reminder, isomerism is about relationships, not individual molecules. So the gloves on the left are isomers of each other, enantiomers of each other, but they are diastereomers of the glove on the right. While the gloves that are enantiomers could hold the ball equally well with each other, if we again imagine holding a ball with a glove or hand that has the fingers backwards, it'd be much more difficult. Diastereomers will have properties that are completely independent of each other in all cases, whereas the properties of enantiomers were only different when we were interacting with something chiral like a glove. To put these isomers in a larger context, any 2 molecules that have different formulas are not isomers. So we have to have the same formula for it to be an isomer. If 2 molecules are different from each other but have the same formula, they are isomers. Isomers that have atoms bonded differently to each other are constitutional isomers. This larger group contains most of the types of isomers we talked about in previous chapters, things like skeletal, positional, and functional group isomers. But if there's no difference in the order atoms are bonded, only the orientation in space, then the molecules are stereoisomers. If they're exact mirror images like your hands, they're enantiomers. If they're not, they're diastereomers. Diastereomers include things like cis-trans isomers, like we saw in alkenes and cyclic molecules, but they'll also include a lot of other molecules as well. So we know that we say that we have enantiomers if we have a complete mirror image, and it has every chiral center flipped, and we'd have diastereomers if we only had a few chiral centers flipped. Practically, this can be really hard to see on any given molecule because we aren't always looking at the molecule from the same direction. If you look at the two molecules shown, it's tempting to think that they might be enantiomers, since every OH that was bolded on the left is wedged on the right. But we're actually looking at two sides of the same molecule, D-glucose. The molecule is just rotated 100 degrees in the vertical axis. There are ways to talk about the absolute configuration of each carbon, but that's both relatively difficult on a large molecule, and biochemists and biologists don't really like it. So instead, we'll use a different mode. Instead, biochemist Emil Fischer, who performed many experiments to understand the differences between carbohydrates, developed a method for looking at every chiral center of a carbohydrate in the same way every time. That way we could quickly compare carbohydrates and see if they're the same or different. We call these Fischer projections. Fischer's rules allow us to look at a carbohydrate really quickly and compare it to other carbohydrates, because we'll always be drawing it the exact same way every time. The first rule is that we always need to put the aldehyde or ketone as near the top as possible. For aldehydes, since they're always on the number 1 position, they will always be at the top. So all aldoses will have the aldehyde at the top. Ketoses will always have the ketone at the 2 position. So we're going to have one carbon above the ketone, and we'll have the ketone right below it. The next rule is that we need to have all the carbons arranged vertically. That usually comes pretty naturally if we're already putting the aldehyde or ketone carbons toward the top. We'll just continue straight down, zigzagging vertically. The final rule is that we always need to look at the chiral centers from a very particular viewpoint. Since carbohydrate chiral centers will always have a carbon both above it and below it, we can look at these chiral centers either so that the carbons above and below are pointed away from you, like is shown from the direction of the red arrow, or toward you, like if we were looking from the other side. We'll always look at each chiral center so the carbons above and below are pointed away from you. In the ball and stick model that's shown in the bottom left, where carbons are black, oxygens are red, and hydrogens are white, that means we'd be looking from the left side where the arrow is pointing. Looking from that side gives us the view in the next picture to the right. So if we were standing where the arrow is, we'd see what the second picture shows. The carbon in the middle is the chiral center that we're looking at. The carbons above and below are pointed back into the screen away from that carbon and away from us. And the side groups, the OH and the H, are pointed more towards us. What we see from this angle can be represented in a 3-dimensional structure with the side groups wedged to indicate that they're coming out towards us, and the carbons dashed to indicate that they're going farther away from us. The Fischer projection assumes this is the orientation that you're looking at the molecule, and so it leaves out the dashes and wedges, simply representing this part of the molecule as a cross. So this is the Fischer projection. To be abundantly clear, this only works if you're looking from a very particular view. You have to have the aldehyde or ketone as near to the top as possible. You have to have the carbons arranged vertically. You always have to be looking at each carbon so the carbons above and below are pointed away from you. If we look at the same chiral center on L-glyceraldehyde, which we see on the top right, the enantiomer of D-glyceraldehyde, and we look from the same angle, we're actually looking at this view. The Fischer projection then will have the OH on the left. Using Fischer projections to draw molecules will again let us quickly compare molecules and see if they're the same, or even how they might be related. By the way, the D in D-glyceraldehyde stands for dextrorotatory, meaning right rotating, because it rotates polarized light to the right, which is one of the main ways that we can see the differences between chiral compounds, between enantiomers. The L in L-glyceraldehyde in turn stands for levorotatory, meaning it rotates polarized light to the left or counterclockwise. But conveniently, L-glyceraldehyde also has the OH pointed left on the Fischer projection. So we can pretend that L actually means left in the Fischer projection, and that'll be more useful to us. When we have a longer molecule, we're going to follow the same rules. The carbon chain is vertical with aldehyde or ketone at top. But each carbon we have to look at from the angle so the carbons are pointed away from us, which means we look from a different side. So the line angle looks like the bottom picture, where the green spheres are representing alcohols because I ran out of red spheres. But when we look at the Fischer projections, we're imagining the molecule with all the carbons pointed back, which is really like looking at the molecule bent into a circle, also shown in the bottom right. The Fischer projection will have 4 chiral centers, and again indicated by each of the sets of red arrows. We imagine looking at the top chiral center from the left side, which you can see over here. Since the OH is wedged, it's coming out of the board, which is on the right when we look from that side. So we see the OH on the right in the Fischer projection. The second chiral carbon, we're looking from the other side. Again, so the carbon chain above and below is pointed further away from us. Since the OH is wedged, it indicates it's coming out of the board. If we were actually standing where that second arrow is and looking at that carbon, coming out of the board would be on our left side, which matches the Fischer projection, which has the OH on the left side. If it's hard for you to see that orientation or to imagine that for yourself, notice that if you arrange the molecules with the aldehydes at the top, every time there's a bolded line, the OH group switches in the Fischer projection. So the OH that's pointed on the left side in this line angle structure over here, well, since it's bolded, it ends up on the right. This OH that's on the right side, since it's bolded, ends up on the left. This OH, which is on the left side again of the Fischer projection as drawn, since it's bolded, ends up on the right. But this OH, which is dashed, stays on the right because it's dashed. Again, using Fischer projections to draw molecules is a really important way for us to be able to identify and compare carbohydrates. If we're drawing a complete Fischer projection from scratch, I always start by drawing out the part that isn't chiral first. I start by drawing out the ketone or aldehyde. Here it's drawn on the right, but it would be equally correct to have it drawn on the left since it isn't chiral. For the ketone or aldehyde, the direction is completely arbitrary. There's a carbon above the ketone, but it isn't chiral, so we typically condense the structure and just write CH2OH. Below the ketone are 3 chiral centers, so we put 3 crosses in our structure. At the very bottom of that Fischer projection, we have another carbon that's not chiral, that corresponds to this. So again, we condense it to CH2OH. The carbon for the C, the 2 H's that aren't shown, and then the OH is the alcohol on the bottom. After we've identified which centers would be chiral, then we can think about what they'd look like. It's important to draw out the chiral centers first, because otherwise it's really common to accidentally interpret the aldehyde or ketone as a chiral center. The top chiral carbon, the top chiral carbon is the one just below the ketone, so it's the one right here. If we look from this side, there we go. If we look from that side, our OH is on our left and flipped from how it is shown in the line angle structure. So we'd see it show up like that. Looking at the next chiral carbon from this side, again, because it keeps the carbons pointed away, again gives us the OH on the left side. This time, since the line is dashed, it's on the same side as it was in the line angle structure. We look at the final carbon that is chiral from the right side again, which puts the OH coming out of the screen on our left. We fill out sides without the H with hydrogen, like this. If we left these blank, those lines would be interpreted as carbons, so this is actually a really important step. This molecule is named L-psychose, which is a fun name, but not really important. The L part comes from the final chiral carbon being similar to L-glyceraldehyde, in that it's pointed left, or the OH is pointed left. And that's actually always going to be true. the final chiral carbon will have the OH on the left for all L carbohydrates, and it will have it on the right for all D carbohydrates. Now try to draw the Fischer projections for the remaining molecules, starting by drawing the skeleton with the crosses for each chiral center, and then identifying the chiral centers as either left or right for the OH. For the first molecule, the carbon skeleton is the same, but the top chiral carbon is flipped. So now it's right, left, left. That's the only one that's changed from the leftmost molecule to the next one. Now that you've seen two of these, what do the next two look like? Again, the carbon skeletons are the same, but the arrangements for the third molecule, L-sorbose, is left, right, left. And then for D-tagatose, which is the rightmost molecule, is left, left, right. So we'd see a different orientation for each of those oxygens. Again, the names aren't important, but notice that the direction of the final chiral carbon's OH determines the D or L part of the name. So we have L every time the OH is pointed to the left, and we have D every time the OH is on the right. Once we know how to interpret Fischer projections, it's easy to see why they're so important. The figure shows only half of the possible 3 to 6 carbon aldoses. The other half are the enantiomers. Remember, that's mirror images of these molecules. At each branch point along this diagram, one new top chiral center is added, while the bottom chiral centers stay exactly the same. This makes everything in a row a diastereomer of everything else in that row. Because diastereomers all have different properties, they all have different names. Notice that every one of the molecules shown has the very bottom chiral carbon, again, the one that has the OH highlighted, pointed in the same direction. They're all pointed to the right. The D versions of the carbohydrates all have the bottom chiral carbon's oxygen on the right side. They are named this way because they are related to D-glyceraldehyde structurally. All D carbohydrates have the final chiral carbon's oxygen, again, on the right side. D carbohydrates are by far the most commonly occurring in nature. These are a few of the carbohydrates that I want you to memorize: the D-glyceraldehyde, D-glucose, D-galactose, and D-ribose. I put them much larger 2 slides from now, so don't worry about copying them from this slide. There is a narrower selection of ketoses than aldoses, since when we move the aldehyde down 1 carbon to become a ketone, that reduces the number of chiral centers on each molecule. The top carbon is not chiral. But again, anything in the same row is a diastereomer, and we only have drawn the D enantiomers for this diagram because they're a lot more common. From these, I want you to memorize D-fructose and dihydroxyacetone, which again are on the next slide. There are 6 carbohydrates that you should know. D-glyceraldehyde, because it's closely related to central metabolites. That's compounds that are formed when you're converting food to energy. I also want you to know D-ribose because of its role in nucleic acids like ribonucleic acid. I'd like you to know D-glucose because it's the most versatile form of energy transferred in our bloodstream, and it's also the monosaccharide that's most present in our diet. D-galactose and D-fructose are also major dietary carbohydrates. And finally, dihydroxyacetone is related to a lot of other central metabolites. For most people, it's probably easier to memorize the structure based on its functional group and length. For example, aldotriose for D-glyceraldehyde. We also can then just memorize the direction of the OH on chiral centers. So for D-ribose, we'd say that it's an aldopentose. Again, that's an aldehyde containing carbohydrate that has 5 carbons, and then that its chiral centers are right, right, right. This converts the structure into a short sequence of words from which you can recreate the structure. Or, you can try to just stare at the screen long enough to burn them into your retina, but I wouldn't recommend it. So if you use things like the functional group and the length, and then the direction of the chiral centers, that can make your life a little easier. Just to hit some of the definitions that we've talked about already in terms of carbohydrates, diastereomers are molecules that have the same connectivity, but can't be mapped on top of each other, so they're stereoisomers, and they're also not mirror images, so they can't be enantiomers. This happens when some, but not all of the chiral centers have flipped direction. On the chart of aldoses, this is the relationship within any of the rows. In biology, sometimes a single chiral center gets flipped. We'll see this right at the beginning of our next section. When only one chiral center is flipped, it's still a diastereomer, but we also give it a more specific name, epimer. Epimers are a specific type of diastereomer. What isn't shown in this image are enantiomers, which reverse every chiral center. So in the bottom right, I took the D-talose, that's drawn in the last bottom right blue square, which is left, 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 right for the OH groups, and then I drew the mirror image right next to it, the enantiomer. Every chiral center in the enantiomer is flipped, so left, 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 right for D-talose becomes right, 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 left for the enantiomer. When we have the L enantiomer of a carbohydrate, it isn't just the bottom chiral carbon that flips from the right to the left. It's every chiral center that's going to flip into the opposite form. Chirality isn't just important for carbohydrates. It's also really important for medicine. On occasion, drugs are sold as mixtures of both enantiomers, which we call a racemate. But often only one of the enantiomers does what we want, and the other enantiomer only increases the chance of side effects. For example, the drug zopiclone, also known by the trade name Imovane, is sold as a racemate in other countries. The chiral center is drawn as a squiggly line to indicate that it could be pointed in either direction. Only S-zopiclone, which is just one enantiomer of the drug, is active. So notice that now we have a specific orientation for that bond. And so only the S-zopiclone version of the drug, which is known by the trade name Lunesta, can be sold in the US. Some drugs are only sold as pure single enantiomers. Ethambutol, which is used to treat tuberculosis, has two chiral centers and is always sold pure. So those two chiral centers where the bold lines are always have to be in a very specific direction. There are a few drugs that can actually convert form. Ibuprofen is always sold as a racemate. Again, that's a mixture of both enantiomers. But only dexibuprofen, this form, is active. Interestingly, both forms are converted in your body to the active form, so it doesn't actually matter that it's sold impurely. That's why we do it. There are many drugs like steroids that have tons of chiral centers, and every one of them needs to be exactly right for it to work correctly. Dexamethasone, which is an inexpensive and common steroid that has been on my slide deck for about 10 years, but it's gained some recent attention as a part of a COVID treatment, actually has 8 chiral centers. Flipping any of them would likely change its activity. It's one thing to say that side effects are more common when drugs aren't enantiopure. That may not be such a big deal if the side effect is a headache, but sometimes this plays out in much more tragic ways. Thalidomide, the drug shown, was given in the late 50s to treat morning sickness. Only one enantiomer, R-thalidomide, actually was active. However, the enantiomers easily interconvert in the body, and the other enantiomer causes birth defects, affecting about 10,000 fetuses before it was outlawed, and about 40% of those didn't survive until birth, and many of the children that did survive ended up with birth defects. After thalidomide, the US made drastic changes to how drugs were produced and tested. All this to say, we talk about chirality along with carbohydrates, but this is also something that has a drastic impact on how we do medicine.